In this lesson, we learn about the cellular respiration. All right, so let's take a look at the three stages of cellular respiration. We're going to start with the first stage, which is called glycolysis. So glycolysis begins with one single molecule of glucose, and that glucose is a 6-carbon compound. Now, it's important to note that it is a 6-carbon compound because as these molecules go through the Krebs cycle, we're basically going to count the carbons, and that's going to help us kind of understand this whole process as we go through here. So again, glycolysis begins with this one molecule of glucose, a 6-carbon compound, and you see that right here at the top. And then we basically break that glucose down into 2 pyruvic acids, and they're right underneath that blue arrow. And basically, those 2 pyruvic acids are then going to go into the next stage, which you can see down there is the Krebs cycle. But in the process of converting this glucose into 2 pyruvic acid molecules, we do get a little bit of energy out of that endeavor. So that's kind of nice. And so glycolysis, the name for this stage, is based on the fact that it's breaking down, or it's lysing. Lysis means to split. It's splitting that glucose. Okay, and we end up with 2 molecules now, each having 3 of the carbons from the 6-carbon glucose. And again, we call these pyruvic acids. So how many carbons does each molecule of pyruvic acid contain? Well, it's 3. Why do I keep saying that? Because it's important to keep track of everything that's going on here. All right, so we've got glucose up here at the top. We've got the pyruvic acids here in the middle. Okay, we have an input of 2 ATP and an output of ADP. That means that it actually took some energy for the glucose to get split into pyruvic acids, but also some energy is released, and that converts some ADP into ATP. Okay, now the ATP is kind of the currency of the cell in terms of energy. It's the molecule that contains the energy that cells use to drive other cellular reactions. So the whole point of cellular respiration is to produce ATP molecules. Now we also see that there are some NAD pluses involved here too. But let's back up. Let's kind of take this step by step. So we've got the 2 ATP that go into the reaction, and the result is they're kind of used up. Some of their energy is drained away by splitting off a phosphate group from the end, and now we have adenosine diphosphate instead of triphosphate. Okay. And so it's a bit of an investment, actually, if you think about it. So we invest 2 ATP into the process, and we actually get 4 ATP out of the process. We see that right here. And as a result, we end up with a net gain of 2. So we end up with 2 more ATP than we started with. We put 2 in, we get 4 out. 4 minus 2 is 2. So that's kind of, you can think of that in an investment model sort of way, where it invests 2 ATP to get 4 back. So they've increased by 2. Okay, so it's this energy input right here that actually drives the start of the reactions that are going to eventually break the glucose into the two pyruvic acids. So as those reactions get kicked off with the energy input of ATP, we see some other things happening as well. Okay, we see NAD plus reacting and picking up some high energy electrons because it is an electron carrier, a high energy electron carrier, and it forms NADH. And that NADH then is going to eventually move on to the electron transport chain, which is stage 3 of cellular respiration. We also have some ADP getting involved right here, and that gives us 4 ATP. Okay. So once all these reactions occur, and you're only seeing some of the reactions, these are the important ones. This is what we really need to know. We need to know about the 2 ATP that go in and the 4 ATP that come out, and the NAD plus that goes in, 2 of those, and 2 of them, 2 NADHs come out, and eventually we end up with the 2 pyruvic acids. And those pyruvic acids are then going to move on to the Krebs cycle, and they're going to be used to generate some other products that are going to help with the electron transport chain. Okay, so what are the primary products of glycolysis here? Because that's what we really need to kind of keep track of. Well, we find that we have 2 NADH, and we get 4 ATP molecules, and we get 2 pyruvic acids. Okay, and again, those NADH, they're going to head on down to the electron transport chain. But before we get to the electron transport chain, let's talk about the Krebs cycle. The Krebs cycle is the second stage of cellular respiration. And it's important to note that this cycle is essentially a series of chemical reactions. It mainly involves carbon compounds. Now, the element carbon is pretty remarkable because it has the ability to really easily combine with other carbon atoms. It can also combine with all sorts of other atoms as well. And the result is that it can form a tremendous number of different carbon compounds in living things. So while we look at the carbons or the Krebs cycle, we want to kind of keep track of those carbons because the number of carbons in the molecules involved are going to keep changing. And it may seem at first like the Krebs cycle is extremely complex. Well, fact of the matter is, it is very complex, but we're not going to delve deeply into every single reaction that occurs in the Krebs cycle. We're going to focus on the things that are really the most important parts of the Krebs cycle. And one of the ways to kind of simplify the Krebs cycle so it's understandable is to really just kind of follow the carbons, count them as we go. And as we do that and we count those changing carbon compounds, it will make this cycle a little bit easier to understand. Okay? Now, the pyruvic acid that we get from the glycolysis stage is going to get broken down into carbon dioxide in the Krebs cycle. And you see that carbon dioxide here as a product coming out of the Krebs cycle. So let's zoom in a little bit deeper. And what you'll notice is that there are a number of molecules on here. It's starting to look a little more complex already. But what you're going to find is that a lot of these compounds are the same. So we see a lot of NAD+, a lot of NADHs, we see ATP, we see FADH2, and of course we see the carbon dioxide. In fact, here they are all circled for us, so you can see them. And if we zoom in a little bit, you can see we got NADH, we got a carbon dioxide over here, we got another NADH, and we've got another carbon dioxide here, another carbon dioxide here. We've also got another NADH, Another NADH. Oh, there's some ATP. That's fantastic. Oh, there's an FADH too. So really what we're seeing here is a lot of the same thing being produced, okay, at different points during the various chemical reactions. Now, again, there's a lot of chemical reactions going on in here, but we're just focusing on the important things. Now, why is NADH important? Well, it's important because it provides electrons, energized high-energy electrons, for the electron transport chain that we're going to get to later. So that's why the NADH and the FADH2, which is also an electron carrier, why these are important, and that's what we're going to focus on. Of course, the ultimate goal of cellular respiration is to produce ATP. So obviously, we're going to highlight the ATP as well. Now, as far as the carbons go, and you can see there's 3 carbons produced from the 1 pyruvic acid molecule entering into the system. That's important too. When we exhale those carbon dioxides that were generated during cellular respiration. The plants then, as we exhale, those plants can absorb that carbon dioxide from the atmosphere that we've exhaled and other living organisms have exhaled, and they can use that CO2 to build their sugars during photosynthesis. So it's a nice give and take relationship there. Now, one other thing to note before we move on is that glycolysis produces 2 molecules of pyruvic acid from the 1 sugar, remember? Now, the Krebs cycle is going to go through 2 revolutions or 2 cycles, one for each of those pyruvic acids. So for each sugar molecule or for each glucose, the Krebs cycle is going to do 2 turns, okay? Now, here we have another kind of a blow up of the first part of the Krebs cycle, where we have the pyruvic acid moving into the inner space of the mitochondria. So it passes through both the outer mitochondrial membrane and the inner mitochondrial membrane, where it begins reacting. And as it reacts, it actually loses a carbon dioxide. You see the carbon dioxide here. And now we have a molecule that only has 2 carbons. So remember, we're counting the carbons. So each pyruvic acid has 3 carbons in it. It gives off 1 carbon to a carbon dioxide, and now it's a 2 carbon atom or molecule, and it's going to interact with coenzyme A, that's a CoA here, to form acetyl-CoA. Now, before it becomes acetyl-CoA, it goes from being a pyruvic acid to being an acetic acid. That's what this 2 carbon molecule is. The acetic acid then interacts with the coenzyme A to become acetyl-CoA. So just keep in mind, it goes from pyruvic acid to acetyl-CoA. There's a couple things that happens on the way. The important thing here is that we get a carbon dioxide released, and we also can convert an NAD plus into an NADH. That NADH is going to carry hydrogens and charged electrons down to the electron transport chain, which is stage 3. We're going to get to that here in a little bit. All right, so again, we're going to just count the carbons as we go through. We go pyruvic acid, we lose 1 of those carbons to carbon dioxide, we form some NAD plus into NADH, and those two atoms of carbon, the acetic acid, are going to interact with coenzyme A to become acetyl-CoA. Now, the acetyl-CoA is still a 2 carbon molecule, but it has a coenzyme A attached to it. That's going to enable this acetyl-CoA to actually attach to this 4 carbon molecule. And that 4 carbon molecule is now going to be known as citric acid. Citric acid has how many carbons? Well, it's going to take 2 carbons from the acetyl-CoA, both of them. It's going to take all 4 of these here. So 4 plus 2 equals 6. So we should have 6 here if we count them. 1, 2, 3, 4, 5, 6. 6 carbon atoms in the citric acid. So we're able to keep track of all of our carbons so far. Okay. Now we're down one because we released one as a carbon dioxide, not to forget that. Okay, so we have this 6 carbon atom, or the 6 carbon atoms in this molecule of citric acid, and we're going to now follow the citric acid, which is right here, through the cycle. So the first thing that happens is we lose yet another carbon dioxide right here. So now we have not a 6 carbon molecule, but a 5 carbon molecule. Okay, if we count them up, 1, 2, 3, 4, 5. We also, in the process of creating that CO2, just like up above, when the pyruvic acid was being broken down, this molecule of citric acid also releases CO2 as it loses a carbon, and it also loses some hydrogen to this NAD plus along with some charged up electrons. That NADH now is going to be able to head off to the electron transport chain, just like this NADH up at the beginning also moves down to the electron transport chain. Okay? So our carbon molecule continues through the cycle, continues going through reactions, and as you can see here, we now have another carbon dioxide being removed. So now we've gained 3 carbon dioxide molecules, and our 6 carbon citric acid is now down to only 4 carbons, 4 carbon atoms in this molecule. We also have generated more NADH from NAD pluses. And again, the NADH, just like before and just like here, is going to move on to the electron transport chain. So while these reactions are taking place, we're losing CO2, we're converting NADP plus into NADH. We also convert some ADP into ATP by adding a phosphate group to the end of that ADP. Now it's ATP. Okay, so we're starting to generate some ATP in the Krebs cycle as well, and that's important because, again, the whole point of the cellular respiration is to generate ATP. Now, the bulk of the ATP is going to be generated in the next part of the process when we get to the electron transport chain, which we're almost to. But just a couple more points to make here. We now have this 4 carbon molecule. Well, if you remember up here when we were talking about the pyruvic acid breaking down into acetic acid and then reacting with coenzyme A to make acetyl-CoA, that acetyl-CoA then merged with a 4 carbon molecule to form citric acid, right? Well, watch this. Citric acid has 6 carbons. It loses a carbon dioxide, so it loses 1 carbon atom. Now it's a 5 carbon atom. It loses another carbon dioxide, so it loses another carbon. Now it's a 4 carbon molecule again. And look at this. This 4 carbon molecule ends up going right back to the beginning of the Krebs cycle to interact with another acetyl-CoA molecule to become citric acid once again. So it goes from 4 to 6 to 5 to 4. And then back to 6, and then to 5, and then to 4. And then it just keeps going, cycling back and forth. And along the way, the beauty of this cycle is that it's producing more carbon dioxide, it's producing more NADH, it's producing a little bit of ATP, it's producing more FADH2, and this is going to drive the most important phase, although they're all important because they all kind of rely on each other, at least indirectly. But when we get to electron transport, we're going to see a tremendous amount of ATP generated thanks to all of this NADH, FADH2, and the electrons that were generated for the electron transport chain during Krebs cycle. So glycolysis provides some fuel for the Krebs cycle to work. The Krebs cycle provides electron carriers for the electron transport chain to work. So they kind of all rely on one another. Okay, so again, counting the molecules of carbon, or counting the atoms of carbon, we lose 2 more carbons, and now our molecule of 6 carbons is now back down to 4. and it's going to basically work its way back around to the beginning. All right. So let's take a look, before we move on to the electron transport chain, let's take a look at the production of ATP. Okay, so if you recall, there are 2 molecules of pyruvic acid produced during glycolysis before Krebs cycle from the 1 glucose molecule, right? So glycolysis produces 2 molecules of pyruvic acid from that 1 glucose. And so knowing that, we then can figure that each molecule of glucose is going to cause the Krebs cycle to go 2 times because of those 2 pyruvic acids, which I've mentioned before. Now, how many ATP molecules would be produced from 1 molecule of glucose? Well, here we have basically 1 molecule of ATP being generated from the one of the pyruvic acids. But the second pyruvic acid from that glucose is going to enter the system as well and go through its changes and produce another molecule of ATP. So from 1 glucose, we actually get a generation of 2 ATP during the Krebs cycle, thanks to glycolysis. All right. So let's also take a look at how much NADH and FADH2 are formed. So these are formed from the molecules NAD plus and FAD. And you see all of these combinations, all of these reactions are circled here on screen. And basically we have 5 places for FADH, or I'm sorry, for NAD plus to turn into NADH. We got 1, 2, 3, 4. Sorry, 4 places. And then the 5th place would be the FAD converting into FADH2. Okay. So these molecules of NADH and FADH2 are all being driven. The reactions that form those are being driven by the energy that's being released when those carbons are being broken off of the citric acid, or the pyruvic acid in this case up here. But down here it's a result of the carbon being removed from the molecule. So either way, it's a release of a carbon produces some energy that can drive some of these other reactions to form these electron carriers. So that brings us to the 3rd stage of the cellular respiration, and that's called electron transport, or electron transport chain. This is the 3rd stage of cellular respiration, and it is somewhat familiar to you already because the photosynthesis also has an electron transport chain, 2 of them. All right, so here we have electron transport, and the two primary products that are created during electron transport is, of course, energy. That's the big one. That's the main goal here. But we also have an output of water. But the electron transport chain is unique in that it requires oxygen. The other 2 stages, glycolysis and Krebs cycle, don't require oxygen, although indirectly Krebs cycle does require oxygen, because if the electron transport chain doesn't function, then the Krebs cycle stops as well. So our overall products of the electron transport are energy and water. Let's dive in a little bit to this. And remember that the NADH and the FADH2 are byproducts of glycolysis and the Krebs cycle. We get some of those molecules from both of the first 2 stages. And we see that these high energy electrons are going to come in and join the electron transport chain party. So we're looking forward to that arrival. And basically, they're going to bring with them some high energy electrons, which is going to be important to the process here. So one way to organize information in a long reading passage is to create a graphic that represents that information. So in this case, the reading describes the electron transport process and ATP synthesis, which can be represented by a flowchart. It's kind of what we have here in front of us. Flowcharts are very good for looking at a process and how things move from one place to another. So hopefully you're kind of being able to follow this overall flow chart, and then when we kind of zoom in, you can kind of see the more of the detailed flow chart. So the electron carriers are going to come into play here, and they will move across these molecules that are part of the electron transport chain in purple here. And the yellow arrow just indicates the flow of these particular molecules. So the NADH starts at the beginning. The FADH2 starts at basically the third of the molecules in the electron transport chain. Not really that important. I don't even know why I'm telling you that. Just to point out, I suppose, that they don't both start from the very beginning. All right. So, in the process of these NADH and FADH2 molecules coming into the electron transport chain, what's going to happen is they're going to go through some reactions, and those reactions are going to break off hydrogen ions. These hydrogen ions, because the NADH and the FADH2 are going to release some energy in the process of being converted back into FAD in this case, or NAD+. That's going to create some energy that's going to basically draw these hydrogen ions against the concentration gradient from an area of lower concentration to an area of higher concentration. Normally, it would go the other way, and we're going to find out that it does go the other way eventually. But these NADHs and the FADH2s provide the energy to basically force the hydrogen ions across that membrane into the space between the two membranes of the mitochondria. And so we get a large supply, a high concentration of hydrogen ions in between those membranes. That's going to become important a little later. Now, what is formed when oxygen that comes into play here? What is formed when that oxygen combines with hydrogen atoms and some of the electrons? Well, basically what happens is we get water. So if we look at the equation for the electron transport chain, we see we have, and I will zoom in, we've got some hydrogen ions. We've got 4 of them here. Then we've got a molecule of oxygen, so there's 2 oxygen atoms there. Then we have some electrons, thanks to the NADH and FADH2. Also, the hydrogens are thanks to the NADH and FADH2 as well. And the result of that is this reconfigures or recombines into 2 molecules of water. So we got 2 hydrogens here, 2 hydrogens here, another 2 hydrogens over here, 2 hydrogens over here. Remember, they have to stay balanced, same amount of atoms on both sides. We have 2 oxygens here. We've got an oxygen here and an oxygen here. That's 2 on the other side of the equation. And of course, each one of these molecules is going to pick up a couple of electrons each. Okay, so everything is accounted for. All right, so how do the electron carriers use the energy that's generated by passing those electrons down the electron transport chain? Well, the answer to that is they use the energy to transport those hydrogen ions across the inner mitochondrial membrane, as I was pointing out before. They use the energy to transport those hydrogen ions into that space called the intermembrane space in between the 2 membranes. And you see those hydrogen ions passing through right there. I just circled them in red for you. All right, so the inner mitochondrial membrane also has these enzymes that are called ATP synthases. We did learn about these when we learned about photosynthesis. So the same process is going to happen here, but interestingly, we're going to generate a tremendous amount of ATP as a result. So the ATP synthase, this nice big molecule here, is going to basically be a pathway for those hydrogen ions, which are in high concentration down here now. They're going to basically zoom up through that ATP synthase. That kinetic energy is going to cause a mechanism inside the ATP synthase to spin, providing energy to attach a phosphate group to the end of this ADP, resulting in the generation of ATP. Now, this is going to happen a bunch of times as all of these hydrogen ions keep moving across. More and more ATP is going to be generated. Okay, so basically each pair of high energy electrons that moves down the full length of the electron transport chain, this will provide enough energy to produce 3 molecules of ATP. So we can count that all up and figure out how many ATP are going to be generated, but we're not really going to do that. But just know that for each electron, energized electron, we get 3 ATP. Okay, so enough energy from one of those electrons is enough to generate 3 molecules of ATP from 3 ADPs. Okay. All right, so we're going to take a look at cellular respiration as a whole. And as I've told you before, the most important part of cellular respiration is the generation of ATP molecules. So we were counting carbons before, but now we're going to count the ATP. We're going to see how many ATP we end up with. So during glycolysis, when the glucose is getting split up, we're going to gain 2 ATP. Now remember, that was because we put in 2 and we got out 4, but it's a net gain of 2 ATP. Okay. The Krebs cycle is going to produce another 2 ATP. It's also going to produce CO2. But then we've got the electron transport chain. The electron transport chain is where the magic happens, my friends, because the electron transport chain, thanks to all those NADHs and all those FADH2s, are going to be able to generate 32 ATP. That's a huge jump. So you can see why the electron transport chain is very important to cellular respiration, because that's where the majority of our ATP come from. Now, if we count those up, we come up with a number that sounds like 36, because 32 plus 2 is 34, plus 2 more is 36. So you might say cellular respiration produces 36 ATP from each pyruvic acid that goes through the system. However, it's not exactly robotic in that sense, so it doesn't produce 32 ATP in the electron transport chain every single time. It kind of fluctuates a little bit, as organic things do. Organic things aren't precise. They work well enough to provide life-sustaining services, but we literally have to kind of broaden our horizons from 36 specific to more of an ATP range. And that range can be anywhere from 30 to 42. Okay? 30 to 42 ATP are generated during cellular respiration. Okay? So 36 ATP, that's pretty reasonable because it assumes 2 ATPs for every FADH2 and 3 ATPs for every NADH. And usually they lose about 2 ATPs due to the cost of transporting that NADH produced in the cytoplasm into the mitochondria. So anyway, suffice it to say, if you were to say 36 ATP are generated during cellular respiration, I would mark that correct on a quiz or an exam or homework or whatever. But if you were to say somewhere between 30 and 42, that would also be correct. And that, my friends, is cellular respiration.